morning to all sitting in this room, and myself Koel Mukherjee, and my topic is a brief study on liposome-based drug delivery model and the interaction of metallic nanoparticle and protein against cancer in zebrafish. And the presentation, what I am trying to state here is half of the experimental study and half will be the in silico study. So before coming to the main frame, actually three main points which my presentation will follow up. One is the cancer, that we well known about it. It's a very important and famous disease, and it is many experimental groups are working on that. Liposome-based delivery system, as my work based on the drug delivery through liposome-based, so that is the one approach nowadays is going on to make the drug available to the target specific areas for with the liposome coated for some good results. And third is the nanoinformatics, where we are trying to talk about the information technology, the application of the analysis in silico studies related with the nanomaterials, nanoparticles with the proteins which are involved in the cancer. So if we are just go through of overall of my methodology, so there are some sections. The nanoparticles, mainly we have concentrated on zinc oxide nanoparticles, which we have biosynthesized some 2 plants. And as it is, the main experiment was done through liposome base, so liposome encapsulation we did with 3 different concentrations of zinc oxide nanoparticle, which was T1, T2, T3. There was a positive control where we took only the drug molecule and the fish feed, and there was a negative control where the blank liposome was there with the fish feed. And all these 3 we applied on the zebrafish, on which already through the benzene we have made the cancerous growth in the benzene in the zebrafish for tenure of around 15 to 20 days. And after that we saw some cancerous growth, some abnormalities in that structure in some different parts of the organs. And after that we started the treatment with T1, T2, T3, positive control, negative control. And then we did some histological tests and in silico studies on it. So this was the results of the nanoparticle which we have synthesized, a zinc oxide nanoparticle through some FSM. It was there. That was the elemental studies analysis, that whether the zinc oxide or the zinc material is available. And this is the particle size where we got the zinc nanoparticles within the 100 nanometer of scale. So after we designed the liposome and synthesized the liposome, we tried to coat the nanoparticles with liposome also. And we again did the FSM test actually, and those rounded yellow color that shows that there is a liposome structures, round structures with the nanoparticle. And to confirm it, we did again the elemental studies where with the zinc actually we got the carbon peak also, which states that the liposome with zinc is there available. So after the fish or the zebrafish model, fish were treated with the benzene to get the carcinogen and to make the cancer's growth. After a certain time, we saw that the kidney, intestine and liver, in those 3 parts of the body, there was a abnormalities and some reddish color and different growth-like structure. So it was confirmed that the cancer has induced in that zebrafishes. And we, how many fishes were dead in that stage of days, and how many fish survivabilities were there, and we got that there was more than 50% of fish survivability throughout the experiment. So these are the histo tests for those 3 organs. Mainly we try to focus on the intestine, kidney and liver, but the intestine picture, that was the cancer induced, where we saw, the picture is not very good, but we saw that there is some nanoparticle accumulations also, and there are some disruption of the figures, like those things. And then when we get the T3, that the highest concentration of nanoparticle, they try to again make this villi-like structure. This was disrupted here. In the T2 also, it was little bit of starting. T1, it was very much that positive control, these structures were very much disrupted. So that means that the cancer was there and it was disrupted, and with the treatment of T1, T2, T3, gradually again the villi like structures can be seen. So this was the intestine. After that, the liver also we got the same thing. Like here, different type of cellular degeneration was happened, and for all the organs we saw that T3 is the best concentration where the cells were again regenerating actually. Kidney also we saw the same thing. So this was up to here we were talking about the experimental study to confirm that whether with this cancer and from the Beijing only the cancer is happening. We did one ICP-MS test to find that the water quality on which the zebrafish was surviving, that in water also we have different minerals. Whether that minerals is affecting for this regeneration or actually the treatment what we are giving, that is making the regeneration. So we saw that the iron, magnesium and manganese, these all data actually the value, it is falling not within the cytotoxicity level. It is less than that. That means that these metals are not actually from the water. These metals or the regeneration process happen. That is happening from the zinc oxide nanoparticle which was delivered to them. So now coming to the nanoinformatic studies. So we focuses only on the three organs, kidney, liver and intestine. And the table was very big. I have given only a small part of the this table. So what we thought that we will take or we will go through of the different cancer pathways, like cell cycle, MAPK or the metabolic pathway. So different pathways on these three organs where different proteins are involved. From there we picked two proteins for this study. Among these three proteins actually the HGFA and the TP53, these two proteins were not having any PDB structure, so we modeled the structure through modeler and the dope score, we got the best dope score that we are providing. So after getting those 6 protein structures, we tried to